Stanford University.
So you want to just go ahead and fire
You're
this
on.
up? Okay.
Absolutely.
Okay, I'm not, this is a long talk. I'm not going to do all of it. I'm going to do about a third of it, just to kind of get you up to speed.
Who in the room knows what a microRNA is? Okay, now you know what you're dealing with.
Okay, good place to start. Okay, the first thing I want to start with is this quote from Thomas Kuhn's Structure of Scientific Revolutions. He defined a scientific revolution as an episode in which one paradigm is in whole or in part replaced by an incompatible paradigm. And the paradigm that I'm talking about is what has been known since the 1960s as the central dogma of genetics, and that is that DNA is transcribed into RNA, which is translated into protein. This is a one-directional phenomenon, and DNA is the master molecule of the cell. Now, that was the justification, the rationale behind the genome projects, and everything else that has gone on in molecular biology since the 1950s. The first chink in that armor, the first time it started looking like this may not be always true, was the discovery of reverse transcriptases, which were discovered in RNA viruses that would reverse transcribe RNA back into DNA. We now know that there are endogenous reverse transcriptases in virtually everything. What that led to is the beginning of thought that, in fact, a lot of what's going on in the cell with RNA may not be as simple as we thought it was. The central dogma says DNA is the master molecule, RNA is the messenger boy, and protein is the effector. The sole purpose for RNA is simply to carry the genetic message between the nucleus and the ribosomes, and some of the ancillary stuff that goes on in the cell for transfer RNAs, ribosomal RNAs, and things like that. They're structural, they're functional, they're uninteresting, and in fact, I heard a comment from someone who said that when they were in graduate school, this would be in the mid-1980s, late 1980s, that one of their professors said that RNA is totally uninteresting, we know everything there is to know about it. End of discussion. Well, that reminded me a lot of a congressman in the United States Congress in the late 1800s who brought a bill before the House of Representatives to shut down the patent office because he said that everything that could be invented had been. All right, so put that in your iPod. So what we now know is that one of the biggest surprises of the Genome Project, we believe, in fact, we actually had pools. We actually had money that we were throwing around in bars at conferences about how many genes there would be in the human genome. 100,000, 120,000, 130,000. At the end of the day, we've got about 23,000 genes in this enormous genome, 3.3 gigabases of DNA. Only about 1.5% of that DNA is transcribed into RNA that is subsequently translated into protein. Whereas what we found out recently over the last few years is that in the case of our genome, or in the case of the plant Arabidopsis, or the worm C. elegans, or Drosophila, that a substantial amount of the genome is actually transcribed into RNA, but a very small part of that RNA is ultimately translated into protein. In the human genome, it's about 78% of the entire human genome is transcribed into RNA, but less than 2% of that codes for proteins. So what that means is that there's an awful lot of RNA running around out there in the cells, and we have no idea what it's doing. So people started looking. And of course, we knew about the housekeeping RNAs. We know things, transfer RNAs, ribosomal RNAs, small nucleolar RNAs, and all these other things that are basically messenger servicing and structural servicing, protein or nucleic acids. There were some non-coding regulatory RNAs that were known. These are large species like Exist. Exist is an X chromosome non-coding RNA that's responsible for X chromosome inactivation and chromatin remodeling. It's about 7,000 bases long, which by RNA standards is it's an enormous non-coding RNA. But what we didn't know about, when you look at all of this RNA that's known in the bacteria, the archaea, and the eukaryotes, and you've got all these things, these are involved in the spliceosome and things like that, those are structural. Then along comes this guy, the microRNA. And in just a couple seconds, I'm going to go on and explain to you and show you what a microRNA is. But what we've also found is it's not just microRNAs that are in this previously totally unsuspected universe of these very small regulatory RNAs. The family's gotten actually pretty large in recent years. There are things called endogenous silencing RNAs that are about the same size as microRNAs. These are called Pee-wee interacting RNAs because they interact with a specific type of Argonaut family protein called a Pee-wee protein. These are repeat associated siRNAs. These are from Drosophila. They're the same thing as piRNAs in vertebrates. 21U RNAs, again, they're piRNAs except they're from C. elegans. And then there's these really oddball things called transacting siRNAs. So there's this family of these small non-coding RNAs. These things are about 21 to 31 nucleotides long. They are extremely powerful and they are extremely abundant. And until less than 10 years ago, we didn't even know they were there. Mostly because we threw them out. RNA was a contaminant. It was uninteresting. We threw it away. So in the world of non-coding RNAs, this is where we are. We are at the tip of what we believe is a very large and very complicated iceberg, and we're now starting to drill down into it. If you look at the literature in the past 2 years, go into PubMed and type in microRNA, what you're going to see is paper after paper after paper after paper, 5, 10 a week, looking at the role that these small RNAs play in disease. Or you're going to look at the role they play in plant metabolism, primarily in plants of economic importance. What you're not going to see is a lot of papers asking questions like, where'd they come from in the first place? How did they actually come to be? Why do they do what they do and how they do it? So that's where my interest is. And so I'm going to spend just a couple of minutes telling you how these things were discovered. In 1993, Victor Ambrose, when he was still at Harvard, they were screening C. elegans for mutations that would disrupt developmental stages in the worm C. elegans. They found one particular mutant that completely disrupted larval stage 1 to stage 2 transition, and they called it LIN4. But LIN4 didn't code for a protein. Instead, it produced this 22 nucleotide long non-coding RNA. They went on to find that LIN4 actually, what the function of this non-coding RNA was to suppress the expression of another gene called LIN14, which is crucial in the progression from larval stage 1 to stage 2. When LIN4 suppressed this, the worm didn't proceed to the next developmental stage. Moreover, what they found was that this 22 nucleotide sequence that LIN4 produced bound to 7 different sites in an antisense orientation in the 3' UTR of the LIN14 message. So what it was doing was suppressing translation and not allowing the LIN14 message to actually be translated into protein. Between 1993 and 2000, that was the only example of its kind. And people, believe me, everybody said, it's a worm thing, it can't be that important, don't worry about it, or we'd have found more of them. And then in Gary Ruvkin's lab, which was actually just down the hall from Victor's at Harvard, they found a second one in C. elegans. This one they named Let7. It was also, this is its sequence, it's a 22 nucleotide RNA, and it functioned exactly like LIN4 did. It bound to several sites in the 3' untranslated region of a specific message. It bound in several places and suppressed its translation into protein. But what was more important was what one of the graduate students in Gary's lab did, and this is Amy Pasquinelli. What she did was that nobody could find Lin4 in anything but C. elegans. So she pulled together a bunch of RNAs. This was in the days you were cloning by phone. You just call somebody up, say you got some RNA, send it to me. And what she found was in all these organisms, she found Let7, the same 22 nucleotide long RNA. And not only that, it was absolutely conserved. And you're looking here at 600 million years of complete conservation of this 22 nucleotide RNA sequence. Now, when you see that kind of conservation, that's got to be important. But what it kicked open the door to was a cottage industry of everybody saying, well, wait a minute, is it in mine, and are there others? Well, what we know today now is that these non-coding RNAs have been found literally everywhere in plants and animals. There are thousands of them. A really nice paper in 2004, Floyd and Bowman found a particular microRNA sequence that's been conserved for about 400 million years. Mike Axtell and Dave Bartel's lab drilled down into the land plants, found a lot of these microRNAs that were being conserved. And Victor got to name them because he discovered them, called them microRNAs. Okay, very quickly, where do they come from in the genes that we know? They really they don't look like genes. That's why we missed them in the first place, because they don't have the classic sort of promoters and things like that classical messenger RNA protein coding genes have. But what we do know is that there are primary transcripts from RNA in the nucleus. They're mostly RNA Pol II transcribed, and they're capped, they're polyadenylated, so they look like messenger RNAs, except somewhere in that primary sequence, there is a region of 60 to 100 to 110 nucleotides that folds up into a thermodynamically stable hairpin. And that is the primary, or the precursor microRNA right there. When this hairpin forms, it induces two proteins, DROSHA and DGCR8, which then clip this hairpin out of the transcript. It then passes it off to another protein called exportin 5, which then sends it in from the nucleus into the cytoplasm. Once it gets here, it's a double-stranded RNA. The cell recognizes that, eukaryotic cells recognize double-stranded RNAs as bad. It immediately induces the enzyme dicer, which is part of the RNA interference pathway, which is a cellular defense mechanism. It cuts out a mature sequence, 22 nucleotides long, this short hairpin, or out of the hairpin, this short double-stranded RNA. This gets bound by another one of the Argonaut proteins, and then it gets put into what's called the RISC, the RNA-induced silencing complex. MicroRNAs, up to this point, are unique. From this point on, they share the complete pathway of RNA interference, which is an ancient eukaryotic cellular defense mechanism. Okay. This is the structure. This is this thermodynamically stable hairpin. There's the stem, the loop. These are this strand here or this strand, one of the two, and there are thermodynamic rules that determine which is which. One of them is the mature microRNA. The other one's called the miRNA star, the star sequence on the other side of the hairpin. The microRNA mature sequence, regardless of which side of the loop it's on, is the one that's most commonly expressed in cells. The star is either not expressed at all or it's expressed at much lower levels.
Question.
Yes?
I must be missing something because I can't see where gene expression is being knocked down.
Oh, that's down here. I'm sorry. The RISC then takes it to the target.
Okay.
The guide strand, the mature sequence, then is released by the Argonaut, and the RISC binds in an antisense orientation to the message.
Okay.
Sorry, I didn't follow that through. Okay, what do we know? Plants and animals all have them. Some viruses also have them. Plants tend to have fewer. Animals tend to have more. where they're located in the genome in plants is predominantly intergenic. About a quarter to a third of animal microRNAs are located in introns. Interestingly enough, the introns in which they are located is not the gene that it regulates. Never, at least as far as we know so far. The sequence conservation, plants are highly conserved in the mature sequence, not very highly conserved in the precursor, moderately conserved in both in animals. We really don't know much about the primary transcripts because only a few of them have actually been completely identified and sequenced. Clusters are rare in plants, common in animals. Everything down the way here, these are cognate proteins. Drosha and dicer-like proteins in biosynthesis in plants. The same logical function is being carried out by similar but not identical proteins in plants and animals. The two big differences between plant and animal microRNAs is in plants it is siRNA-like. There's actual cleavage and destruction of the message. And the binding is in the open reading frame of the messenger RNA in one site, just like siRNAs work. In animals, it's primarily translational repression, predominantly binding in several sites in the 3' untranslated region of the message. So they're very different mechanisms. But the functions and the processes that are affected are absolutely the same. They target transcription factors, they target enzymes, they target regulatory genes because they are involved in regulating cellular differentiation, disease process response, and stress responses. That's what these things do. So until very recently, the conventional wisdom held two things. One, that these regulatory small RNAs evolved with multicellularity. They are properties of multicellular organisms. And the conventional wisdom was they don't exist in single-celled eukaryotes. We know for sure they do not exist in prokaryotes. They are not there because none of the enzymes, none of the mechanism, none of the machinery is there. The other thing was that plants and animals, because it evolved with multicellularity, probably evolved microRNAs independently. And both of them just happen to fall into the same pathways. Then, about a year and a half ago, along comes this wonderful little organism, Chlamydomonas reinhardti. Two groups, one in China and then one in David Balcom's lab in England, found microRNAs in a single-celled eukaryote. This is what they look like. Most of them are sort of classic hairpin structures with a single microRNA, but some of them are real oddballs. Like some of them are very, very large hairpins, multiple microRNAs. There's recently been a paper in a tunicate that shows that they too have these very large hairpins, and in fact say this is the primary microRNA that's coming out of this hairpin. The other microRNAs are being processed by a completely different pathway that we didn't even know existed. We're not even sure if that pathway even exists in Chlamydomonas. It's all too new. Okay, so how far back do these things go? Well, there was a paper last year out of Greg Hannon's group, Grimson, the senior author, or the first author, looking back in phylogenetic space here to a quantiflagellate, this Amphibodon queenslandica, which is a sponge, Trichoplax adherens, which if you've never seen this thing or read about it, do. It's the coolest organism you've ever seen, even though it doesn't have any microRNAs, therefore it's of no interest to me. It is really cool. They call it a primitive pancake. It's a multicellular organism. It only has 4 cell types. And it looks like a big amoeba. And it's just really cool. It's an entire phylum that consists of a single animal. And then the sea anemone, what they found was that the sea anemone has 40 microRNAs, Trichoplax doesn't have any. This sponge has 9, and this quantiflagellate doesn't have any. The problem is that they conclude that, because this doesn't have them, this does, and this does, that Trichoplax lost them. The problem is that when you're dealing with this kind of deep-rooted phylogeny, it doesn't take much to flip these things around because you're really looking so far back in time using organisms that are alive today, using their DNA and their RNA that it could be iffy. But I'm going to skip this part because it's not all that interesting. I do want to show you this. This is one of the microRNAs from Amphimedon, and you can see it looks like a nice big hairpin, just like you see in other organisms like Chlamydomonas. Okay, and this is where I'm going to end. I'm not going to go through the last part of this. This is purely speculative on my part, but this is where my interest is, and actually, unfortunately, the interest in asking these questions and answering them, there's more people in this room than there are people researching fundamental evolutionary questions in microRNAs, so that's a small club. But you start, back what you guys have been talking about, the origin of life, the origin of the emergence of photosynthesis, the emergence of these the prokaryotic organisms, and then the emergence of the eukaryotes. Where did these regulatory RNAs come from, and how old are they? We know that there are regulatory non-coding RNAs in these organisms like cyanobacteria and other prokaryotes. There's not very many of them, maybe 10 or 12, maybe as many as 20 in some species. And they're much bigger. They're 100 to 300 nucleotides long, and they are all induced by some sort of stress. You have to stress the organism to find them. Then, but they're big and they don't form hairpins. So where did microRNAs come from? Well, we're pretty sure that somewhere around between 2 billion and 1.5 billion years ago, eukaryotes emerged. We started seeing eukaryotic organisms, single-cell primitive eukaryotes. Looking across the eukaryotic world, it's pretty clear that RNA interference appeared very, very early in the evolution of eukaryotes, because the machinery is there in all kinds of single-cell organisms that have been looked at for RNA interference. These piRNAs function in suppressing transposon replication, which for a genome is as dangerous or more so than being attacked by an RNA virus, which is where RNA I Camp comes in. This is a defense against RNA viruses. This is a defense against the genome being overrun by its own transposons. So the question is, where did microRNAs come from? We're starting to look in organisms where we have a clue that certain microRNAs come from transposons, and we're looking for the signatures of those transposons. And we've got a set now in Chlamydomonas that, in fact, we're just writing up the paper now, where we've actually identified a group of microRNAs in Chlamydomonas that are associated with a specific ancient transposon, which is the gypsy transposon. It's one of the oldest transposons known. And there are others, there are breadcrumbs that we're following. The problem is that in doing this kind of work, we're looking at organisms, whether it's a single-celled eukaryote or us, are at the same place in their evolutionary history. Chlamydomonas genome is older than ours, but it still evolves, it still responds, it is still at the same point in its history that we are in our history, which is, February 2009. So you have to try to piece together the breadcrumbs of stuff that is much, much, much older and may have degraded over time. So that's what we're doing. We're piecing them together a little bit at a time. But what we think we've got are some clues, some breadcrumbs, that put together the transposon origin. This all got started, and I'll finish with a story about there's a small group of us that are sort of outcasts in either whether we're at the University of Iowa or in my company, where I'm not so much employed as tolerated. But we were sitting around in a restaurant, and I proposed, this was about 4 years ago, I proposed a classic Einsteinian thought experiment. And if you've never done these, and you've never read about the kinds of things that Einstein did, where he did these wonderful thought experiments that he then derived all the equations to explain what the implications of his thought experiment were, it's your warm ancient sea or warm ancient pool. I am a primitive single-celled eukaryote. I'm not Chlamydomonas. I'm not anything that's around today. I have a genome that is small and very basic. I'm floating around in this warm water. What do I need? I need to eat, I need to reproduce, and I need to defend myself. And that's it. Anything else you think of is going to go under one of those three categories. You respond to your environment, you search for food, you reproduce, you search, by fission. That's it. That's all you need to do. Where could microRNAs have come from? You don't need them to eat, you don't need them to reproduce, but you do need them to respond to your environment. MicroRNAs and siRNAs all pass through a double-stranded RNA phase. If I am a primitive genome, where is the only source of double-stranded RNAs? The only source of double-stranded RNAs is transposons. So we think, following the logic of that thought experiment and now starting to search for the breadcrumbs in these genomes, that microRNAs came from transposons originally. They then piggybacked onto the RNA I defense and the piRNA defense mechanisms that were already present. And that's why microRNAs and piRNAs share the same pathway in the cytoplasm. So that's the gist of that thought experiment and where we're going with microRNAs. And I am going to try to very quickly, I don't want to go through, this is all a bunch of stuff that I've done in marsupials, and you really don't need to know about this. I started with a philosopher, I will end with a philosopher. The noted philosopher Tommy Lee Jones in the movie Men in Black. He and Will Smith are sitting on the park bench just after Will Smith discovered the Men in Black and discovered that there were aliens living on the earth. He said, everything people have ever known has been proven to be wrong. 1,000 years ago, everybody knew as a fact that the earth was the center of the universe. 500 years ago, they knew it was flat. And 15 minutes ago, you knew that we humans were alone on it. Imagine what you'll know tomorrow. That's what the small RNA field has done. It has opened up a universe that we didn't even know was there, and has opened up a whole series of evolutionary questions, biomedical questions, name it. And with apologies to the guy who said that RNAs are uninteresting and we know everything there is to know about them, we're not even close. So that's where we are. So, thanks.
Echoes of the RNA world. I know that we're, once again, ran way over. So I
Yes, thanks for being
suggest
patient.
that
Appreciate
people
that.
who have questions for Dr. Devor, why don't you come forward and go ahead and ask them, because this is your one and only chance. And thank you again to both of our speakers and to all of you for staying so late. So remember, we're switching to Men in Space, from Men in Black to Men in Space next Tuesday. So quickly learn if I will send out some more emails. Mercury, Gemini, Apollo.
For more, please visit us at stanford.edu.